Okay, so hello everyone. I think I know all of you by looking at the list of people attending, but I am Elin and I'm a PhD student in Thomas Tegnér's research group. And as Eva said, today I'm going to talk to you about biofilms and Klebsiella. But before we get started on that, let's see that I can. Yes, it works. Uh, I feel like we all know the reason behind why we're sitting here talking today. And I mean, it is this great antibiotic resistant problem that we have. And I would like to, I'm not going to talk too much about it, but I'd like to read this quote by Sir Alexander Fleming from his Nobel lecture in 1945, that the time may come when penicillin can be bought by anyone in the shops. Then there is the danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. And I think this quote is so, I think it really shows that we have known about the risk of resistant development and spread for such a very long time, but also that this dystopic future that Sir Fleming warned us about is now actually here. And that I don't have to tell you about the number of lives lost to infections caused by multi-drug resistant bacteria, or the fact that the silent tsunami that we have been talking about for so long now is actually a roaring wave that I would say have already hit us. So instead of that, I would like to take this time that we have now to discuss the future. And by doing so, presenting the work that I have done so far during my PhD, or more specifically, we're going to talk about my project 3, and also kind of my attempt, I guess, to be part of some kind of solution, hopefully, eventually. So, but I know that we have kind of scattered backgrounds here, which is great. But just to make sure we're all on the same page, I'm going to describe 2 core concepts to understand what I'm actually doing. And that is beta-lactam, beta-lactamase inhibitors and biofilms. So let's start with the beta-lactam antibiotics. I think most of you are familiar with the beta-lactam drugs. They are the most prescribed class of antibiotics in clinics today. And I would say that we have these 4 main classes, the penicillins, cephalosporins, carbapenems, and monobactams, right? But they all kind of work by the same mechanism of action. They enter through the bacteria through these porins, and then they bind to these penicillin binding proteins right here. And the function of the penicillin binding proteins is to cross-link the peptidoglycan chains. And when that is inhibited and cannot happen, the bacterial cell wall will eventually be destabilized, and the entire cell wall will be kind of wobbly and eventually break. And that will kill the bacteria, which when we treat with this kind of drug is exactly what we want. However, as we know by now, bacteria have this extremely annoying tendency of gaining resistance, and there is no exception for the beta-lactam drugs. When it comes to gram-negative bacteria and beta-lactam antibiotics, the most common type of resistance is the production of beta-lactamases. And those are these, we say, hydrolyzing enzymes, which means that they break down the beta-lactam drug before it can reach its target. And there are so many different types of beta-lactamases out there. When I started my PhD, I got terrified because I didn't understand how I should know all the names, and that I would say by now is pretty much impossible. In an attempt to get some structure, there are different classification systems you can use. I would say that the most common one is the Ambler classification system, where we have class A, B, C, and D, and they are divided into these groups based on their amino acid sequence. You can also, and for the sake of this presentation, because this will be important later, divide them into 2 groups based on which mechanism they use to hydrolyze or to break down the drug. So both A, C, and D are what we call serine beta-lactamases, or SBLs. And this means that they have a serine residue in their active sites. And the group that then stands out, and that I would say is the most problematic one right now, is the metallo-beta-lactamases, or the MBLs. And they differ in the way that they instead have 1 or 2 zinc ions in their active sites. So that will be important later, but we will circle back to that. I also have a physician as my supervisor, so I also want to just briefly mention 2 very clinically important groups of beta-lactamases. And the first one is the ESBLs, or the extended spectrum beta-lactamases that we see here. You see they span over almost all classes. And they have the ability to hydrolyze both the penicillins and the cephalosporins, which is extremely problematic. But the group that I would say is the most terrifying one is really the carbapenemases, because these ones, they can break down all of our beta-lactam drugs, including the carbapenems, which has kind of been the last line of resort, the last solution or the last treatment option for a while now. So that is very scary. And here we have KPC, we have NDM, and we have OXA48. So that is scary. However, if you want to protect your beta-lactam drug against these beta-lactamases, you can dose it together with a beta-lactamase inhibitor. And this works by, they enter through the porins as well, and then they bind to the enzymes, kind of keeping them occupied. And by doing so, the beta-lactam drug can sneak past the enzymes still in its active form and reach its target and kill the bacteria, which is awesome. And there are actually quite a few different approved combination of beta-lactam, beta-lactamase inhibitors on the market today. The older ones have kind of a more narrow spectrum. So there was a need for beta-lactamase inhibitors with a lot broader spectrums. And there are a few out there today. However, there are no beta-lactamase inhibitor on the market with activity against the class B metallobeta-lactamases. It's a huge problem because bacteria tend to produce beta-lactamases from all different classes at the same time. So we need something that spans all the different classes at the same time. And when I starting to do my PhD, there were nothing of that sort. However, if you look now at this, my little table, there is a combination right here that spans all of them. And that is aztreonam and avibactam. And this is the combination that I have been working with for many, many years now. And the metallobeta-lactamases, as I say, they hydrolyze most beta-lactam antibiotics, except for the monobactams. And the only monobactam available on the market today is aztreonam. So by combining aztreonam with avibactam, we will have a combination that will work both against the SBLs and the MBLs, which is amazing. And this is actually now finally approved for clinical use since last year. It goes under the name Emblaveo. And I just wanted for fun to show this picture of me and my co-author at ESCMID Global last year. We were standing in front of Pfizer's kind of monter for this new combination drug, and that we were just so excited that is finally will be giving to real patients who actually needs it. And I also want to stress that this is not in any way, shape, or form in collaboration with Pfizer. We were just very, very happy. So that is the beta-lactam, beta-lactamase inhibitor combination that I work with. I would now like us to kind of take a step back and look at antibiotic resistance, like as a field altogether. Because if you think about it, the majority of studies that are performed today are done on planktonic bacteria, meaning that you have a culture where you grow your bacteria where they're like swimming around. But not nearly as many are conducted on bacteria living as biofilms. And that is unfortunate, since, as one of my favorite biofilm researchers once said, biofilms are one of the most widely distributed and successful modes of life on Earth. So let's dig into the world of biofilms just for a moment. So the definition of a biofilms are aggregates of microorganisms, sometimes single, but I would say mostly multi-species, that covers themselves within this slimy matrix of extracellular polymeric substances, or EPS for short. And within this matrix, they adhere to each other and also possibly to a surface. And you can imagine that within this kind of matrix that they build, there is also an accumulation of a lot of different stuff. I mean, you have live cells, you have dead cells, dormant cells, cells with different antibiotic resistance mechanisms. You also have proteins, polysaccharides, extracellular DNA, and also antibiotic degrading enzymes, such as beta-lactamases. And it's all of these components that creates these bacterial communities from where them cells can disperse and relocalize to new places and set up new biofilms. So The life cycle of a biofilms looks like this, and these illustration I have borrowed from Patricia, my biofilm queen in Linus Sandegren's group. So first you have the initial cells that attaches to a surface and or to each other. And if the conditions are just right and suitable for growth, they will start to initiate biofilm formation. And that is then followed by cell aggregation. They're getting more and more cells in there, and they start to produce this matrix. And they will continue to grow and mature until it reaches its maximum cell density. And what is so fascinating about these biofilms that is within the same community, we will have micro environments that are established due to gradients. Gradients such as nitrogen or oxygen. And because these will come from the outside, right? And then they will be consumed first by the cells in the outer layers, and less will penetrate into the deeper layers of the biofilm. Meaning that these gradients will create subpopulations of bacteria where you have kind of the fast growing cells at the top, and you have the more slower, more nutrient deprived cells in the middle. And then finally you have dispersion, and this is a huge problem when it comes to infections, because then cells will start to release from the biofilm, and they can relocalize and infect somewhere else, set up a new biofilm somewhere else. And that is can be very fatal in a patient with an ongoing infection. And that is also why biofilms are such a big problem, because 65, I think the number might be even higher, but at least 65 of all infections are in some way related to biofilm formation. And the scary part is that they can be 10 to 1,000 times more resilient to antibiotics compared to their planktonic counterparts. And that is quite high numbers. But what I also feel is important, because maybe some of you will be super interested in biofilms and start to read about it after my talk. I hope so, at least. And then I think it's important that we make the distinction between resistance and tolerance, because this is widely discussed within the field. And just to make clear that resistance, there are traits that comes with a specific bacterial cell, right? So it's often acquired through mutations or gene exchange. It can be efflux pumps or the production of beta-lactamases. And then we have tolerance. And that is traits that comes, the benefits of living in this biofilm lifestyle, and that will be lost once a cell disperses from the community. And that is very obvious things such as the matrix production, but also things that can be beneficial due to the metabolic heterogeneity we get due to the nutrient gradients. Yes, so in the middle of all that, the Aztreonam-avibactam combination and the biofilm, we have my project 3, my third PhD project. And the aim here was that we wanted to expand the knowledge about the effect of beta-lactam, beta-lactamase inhibitors against biofilms, because there isn't actually that much about that out in the literature today. And from what I have understood from talking to different physicians, that you don't really use this kind of combination against a biofilm infection. So we wanted to test the effect of Aztreonam-avibactam against ESBL and carbapenemase producing strains of Klebsiella pneumonia in a biofilm setting. And since in the Tegnéen group we also work very much with combinations, we also tried to add colistin and fosfomycin into the mix to see if that had an additional effect. And we chose those drugs because previously or other studies has shown that they have worked very nicely against biofilms, both in combinations and as in single treatments. So the method we use is this awesome PEG model, the FlexiPEG setup, that is an in-house model that I think this was developed in the D73 corridor. So you have this kind of lid that fits perfectly over a 96 well plate. And within this lid, you have these pegs that are illustrated right here. And then you inoculate your wells with bacterial suspension, and you just put your lid on top of that. And the biofilm will start to form on these pegs. So we let them form and grow for 48 hours. And then we wash them to get rid of all the cells that have not attached to the biofilm yet. And then we add our drugs. So in our case, then Aztreonam-avibactam with or without colistin or fosfomycin. We let them grow for additional 24 hours, and then we harvest. And this is the part which I think is so incredibly neat, because then you take your peg lid with the pegs and put it on top of a rack with these glass tubes, which I fill with PBS, and then you just push the pegs straight through the lid down into the glass tubes. You don't have to interfere with the pegs in any way. You don't have to disturb the biofilm in any way, and I think that is so easy to work with, right? And then I just vortex them to disperse the biofilm, and then I can plate my suspension to see which viable count I have. How much cells do I have? So we have worked specifically with constructed strains of Klebsiella. So we started with this 80cc strain of Klebsiella pneumoniae, and the rest of them, all of them are isogenic, meaning that they look genetically the same, but they differ in which beta-lactamase they produce. So we have both KPC2, OXA48, and CTXN15. They are SBLs, with KPC2 and OXA48 being carbapennamases and CTXN15 being a very widely spread and problematic ESBL. And then we also have NDN1, which is an MBL, but also a carbapennamase. So this is the setup that we have used. It's quite straightforward. And now when we go to the results, this can be quite hard to look at, so I will break it down. But this is the Astronom avibactam data, and let's start with the KPC2 and CTXN15 producers. So what we do, because when I look at this graph for the first time, I felt like it was upside down, but it's actually, we have plotted log killing compared to the growth control without antibiotics. So the longer this bar is, the more killing the combination or the drug we have achieved. So a long bar is good. So we have Astronom as single treatment, we have Astronom in different concentrations together with different avibactam concentrations. And what we can see for the KPC2 and CTXN15 producers is that Astronom as single treatment has absolutely no effect at all. And this was not very surprising because both CTXN15 and KPC2 are very good at breaking down Astronom in planktonic cultures, and it seems to be the same in biofilm. However, when we add avibactam, then we start to see effect, and it's a very clear concentration-depending effect all over the graph, and it seems to. But the effect of the avibactam seems to cap out somewhere around 16 to 64 milligrams per liter, because as you can see here, we tried these more extreme concentration as well, and that doesn't really do any difference. So we absolutely see an effect of Astronom avibactam in a biofilm setting, and we actually reach a maximum killing of up to -4 logs. And if you're used to work with planktonic cultures, it might seem like, but Elin, they are not entirely dead. That is true, but biofilms are also so incredibly hard to eradicate completely. So a -4 log reduction in growth is actually very, very promising results. So that is super exciting. If we look at the other two, the NDM1 and the OXA48 producers, the results are a bit different. We can see that Astronom single treatment seems to have a great effect, both at the NDM1 and OXA48. For the NDM1, that's not very surprising, since NDM1 has no effect against Astronom whatsoever. It's a metallo-beta-lactamase, but it also seems like OXA48 is quite bad at hydrolyzing Astronom. Avibactam has no effect in the NDM1 producer at all, which was expected. There is no beta-lactamase inhibitor with effect against the MBLs, but we can also see then that avibactam have a little bit of effect in the OXA48. Not as clear, I would say, as in the CTXN15 and KPC2 producers, but a slight effect. But it's also quite clear that Astronom single works quite well. Then these, I would say, are the most fun and interesting results that we saw, because unfortunately, the addition of colistin to this combination did not have basically any effect at all in any of the beta-lactamase producers. The only place we actually saw some kind of signal is in the KPC2 producer, at a very high colistin concentration. So we have colistin at 0, 1, and 4 milligrams per liter. And it's only at 4 milligrams per liter we can see a slight better effect compared to no colistin at all. And this was quite shocking to me because colistin has had very good effect in previous studies that I have looked at, specifically in biofilm. So I am not really sure why we don't see that effect here. And unfortunately, it's the same when we look at fosfomycin, and here we don't see any signal at all. The addition of fosfomycin did absolutely nothing, which is, to me, once again, very surprising and unfortunate. But yeah, that's just how it is. So what we can conclude from this is that aztreonam, as both single treatment and in a combination with avibactam, actually had a significant effect against the biofilm forming strains included in this study, which is super cool and maybe an indication that this combination could be used in patients at some point. I asked my supervisor about this the other day, like, would you actually consider using this? And he said that, I mean, if there is nothing else to do, what do you do? Then you try something. And then this maybe might be something to try. We also had the fact that the addition of colistin to the aztreonam-avibactam combination only had an effect in the KPC2 producing strain, and that was only at a high 4 milligram per liter concentration. And we also have the fact that no additional effect was seen by the addition of fosfomycin whatsoever. So with that, I would like to say thank you all so much for listening. Thank you to my supervisors, my fellow PhDs, old and new, Sandegren Group, especially a big shout out to Patricia for letting me use your beautiful illustrations, the D73 corridor and the gradient tech ladies that we share our lab with. And thank you all for listening.
Thank you, Elin. Thank you all for your applauses and your responses and reactions. I have some questions, and I hope maybe someone else have some questions. But I'd like to start more generally speaking. You know, when you're talking about biofilms, I think it's freaking amazing. You know that. Amazing, not in the sense of good, but that
no, but
that
fascinating.
that so many infections have an underlying cause related to biofilms. So and I don't really ask this thinking that you know the answers. This is more of like a discussion or things that come to my mind when I hear about this. To what extent do you think, first, that doctors know the role of biofilms in infections, and also how much do they communicate about this, if they know, to the patients, in the sense of talking about how difficult it might actually to treat something because of this particular physiology, which I know is, you know, we're amazed by it because of biology, but you know that I love communications. I like how does this translate into what the doctors know, and how much do they talk about this? I mean, you talk to Thomas, and Thomas is an infectious diseases doctor.
Yeah, he's.
So have you talked with him about this?
I mean, it's a very good question. I think it might also be a question that my dear friend and colleague Lisa Faxén has a better answer to, who is actually a physician, right? But I mean, I hope that there is something that they discuss. And I mean, since it's so, as you said, I think the numbers I heard from Patricia last time she presented was that 65 to 85% of all infections are caused by biofilms. So I hope there is something they are knowledgeable about and that they take in. I mean, on the infections board, I am sure that there is something that they discuss. Absolutely.
Will be the infectious diseases specialists that see doctors. But because I draw from my own experience, you know, my mom had a continuous asymptomatic urinary tract infection of sorts. And what it turns out to be, what I think, because the doctors never really fully said, is that many years ago, she had a very serious kidney infection that, well, it was pyelonephritis, right? So what they ended up thinking, and we think, is that the scar tissue that infection made in the kidney, it got an infection by a biofilm that it basically was always producing bacteria. So that's why when they would do a urinary test, they would get a positive for bacteria in the urine, but she was asymptomatic. It's just that it was that biofilm was living there basically
Yeah.
and shedding this bacteria.
Mm,
You
mm,
will get
mm,
it in the test, but they weren't really causing asymptomatic infection, right?
mm,
And it took many years and many different treatments because they will see a positive urine test, so they will give her this treatment and then this other
mm,
treatment and then long-term treatment.
mm,
At some point, she was like, I don't want to take more antibiotics because
mm,
obviously this is not working.
mm,
And then I explained to my mom, well, if it's a biofilm in that kind of scar tissue, maybe there is no irrigation with the blood, so the antibiotics cannot really make it to that particular location.
mm.
So it was many years of wondering what was happening, and in no occasion that I can remember the word biofilm was even like brought up.
Nej.
So I just find it very interesting that, we scientifically know a lot of infections have an underlying cause by
Yeah.
biofilms, and I don't know how much that is actually talked and
I,
discussed.
and I mean, and that's just I mean, and I think that comes back to, and we talk a lot about communication, especially on the podcast, right? But then it's also the question of how do you communicate with a patient? Because I mean, I guess for us, it's the term biofilm
Hmm.
feels very natural, right? But I think for many, when you talk about biofilm in Swedish, you think about watching the cinema, right? So,
Yeah, I didn't think about that in Swedish, but yeah,
mm.
yeah, yeah,
So I don't know. It would be interesting to hear from a. Yeah, now Lisa Faxén is raising her hand. Maybe she can actually add something to this, how you communicate with patients about this, because frankly, I don't know.
I would say that we take into the account that the biofilm might be involved in the infection, but to the patient, we don't use that word.
Yeah.
In other words, we are explaining that he or she might need other types of antibiotic or a longer period of time or so. So, but we don't know much about it. So this research is so interesting and important. But we know, like based on experience and clinical studies, like which antibiotic works better than other.
Mm, mm,
So we often add, for example, rifampicin that
mm,
works well in combination with quinolones, for example,
mm.
when it's prosthetic infection, and so we take into account what we know, but
Yeah, yeah.
more research is needed. But often we don't use the word, so that's why it might be not mentioned so often for patients.
yeah.
But do you regulate the dose in any way when you?
Yes,
Yeah.
yeah. So yeah, doses and types of antibiotics. So it differs a lot if we think that if the patient has foreign material in the body, and we think that the infection is associated to this foreign material, we it changes the doses and types of drug we use.
Yeah.
Yeah,
Mm.
very, very interesting. Thank you for bringing that,
Yeah.
It's
perspective.
so nice to have a physician here who can
Exactly,
answer these
exactly. Yeah.
questions.
So now going into the biology of things, I have some questions. I mean, I understand that you didn't expect to see no effect from the colistin.
No.
Um, so I wonder, have you explored the potential reasons for this in terms of maybe experimental setup or any other wild hypothesis out there of why you are not seeing what other kind of studies have seen before?
No, and that is something I would like to explore. I was actually trying to pitch to my supervisors maybe doing a biofilm time kill, right? To see if we actually have some kind of resistance development during this period that we treat. Because I know that in my group we have worked a lot with the colistin in planktonic time kill settings, and we often see resistance development just during those 24 hours. So I don't know if it can be that since we have treatment for 24 hours, that we have some kind of effect in the beginning, but then they gain some kind of resistance mechanisms and just grow back. I don't know. Because to me, it's very surprising that the colistin does nothing, especially also because colistin have a very different, mechanism of action compared to the beta-lactam drugs. So I thought that it would kind of enhance each other.
But can it be also these other experiments or studies that you've seen are also done in biofilm setup?
Yeah.
Yeah, because I'm thinking, since it's a very particular physical aspect of how colistin works,
Mm,
the physicality of the membrane, right? With
yeah.
with the colistin.
Mm.
If the fact that, maybe it's a different kind of biofilm. Like, do we know
Yes.
what is being expressed in the different kind of biofilms of these studies
And that is a major point, right? That's comparing planktonic cultures with each other are very easy in a way. When it comes to biofilms, there are so many different models. There's so many different ways to grow them, for how long you treat, and and
And
how
is it the biofilm? Is it also in Klebsiella, all these studies?
it differs a lot. I would say that I see many studies in Pseudomonas and yeah, I mean, there's an entire other world with gram positive, like
Yeah,
prosthetic
because
joint infections and such.
where I'm hinting at is that, like the environment that a different species will create to make its own biofilm, it would probably be very different between species.
Yes, absolutely,
And
absolutely.
if
But
any,
I've seen some papers with colistin and Klebsiella biofilms,
oh,
and
okay. Mm.
but I have discussed a lot with Patricia if it can be that you have some kind of interaction with the drug, with some kind of the components of the EPS, and that might be to be an expression of something that will just interact with the drug in a way that.
Yeah, it's like how much can you control for that and how much
Yeah,
these other studies? No, it's a bit like, the quintessential question is the water in the lab different, and that can explain a
absolutely,
lot.
absolutely. Yeah. And that's what
Yeah,
makes this kind of complex as well, right? There's so many different like parameters to take into account.
yeah, yeah,
So it
yeah.
might just be that these biofilms that colistin just don't work.
Yeah, yeah, interesting, very interesting. I think it's even more interesting when your results point you in a different direction than what you expected, in a sense. So,
Absolutely.
or
Yeah,
like
yeah.
philosophy of science, but I think it's interesting because it kind of maybe new doors that you didn't think of will open with that.
Mm.
Someone else has a question for Elin? I have another question if nobody has, and it's also more on the biology side of
Mm.
things. Have you thought about why you get these different results on your beta lactamases, like different producers, like the reason behind it?
I mean, if you compare these results with like planktonic results, it all makes a lot of sense since they have such. And for the NDM1, for the metallo-beta lactamase, for example, we do not expect any effect.
Yeah. That one is it, but the
Yeah,
other.
and OXA48 is notoriously known to be a little less good of a beta lactamase compared to
Okay.
XM15 and KPC2. So I mean, the results in that way, if you look at actual enzymes, are not that surprising. It's
Okay.
just very cool to see that it's translatable from planktonic culture into a biofilm setting.
Yeah. Yeah. All right. Then I understand. I don't know so much about the different
No,
beta.
that's fair.
I know, serine and
Yeah,
metallo
yeah, yeah.
are like dangerous, more dangerous. So
They
yeah.
are very, it is very specific knowledge.
I It's very interesting, that you've been working with Avibacta and Astronam mix for some time, but only last year it kind of came into, being used in the
Mm.
clinics. What is the main indication for it?
Oh, once again, Lisa, do you have input on this? I guess you don't
The
use
main
it
indication for
carefully.
Astrionum avibactam.
Yeah.
Yes, it's infections with, as Elin says, metallobeta lactamase. So, like for example, NDM producers.
So it's all, there's a lot of, like, there's a whole diagnostic first, and it's done as a kind of down, okay, how you say it, like you will change the treatment once you know that it's a.
Exactly.
It's not a first line drug, I assume.
Exactly. No,
Yeah.
usually we start with like maybe carbapenems, then like when we get the results from the susceptibility testing, then sometimes like with a patient from Ukraine nowadays, sometimes it's the only drug that works. So we had like yeah. So yeah. So we use it actually a lot when there is these horrible cases with very resistant strains.
Have we already seen some resistance to it as well?
Yes, yeah, because actually it has been used for a while, but we had the drug ceftazidime avibactam,
Oh, yeah.
and we have used it with the Astrionam. So it was like a combination drug with Astrionam. So that has been used like in India for a long time. So we see some resistance already. So yeah, we need to use it when we
Little
really
as
have to.
possible. Yeah.
Mm.
Yeah, I understand. I mean, it's very interesting to see that, some combination that was used more empirically than anything, then it actually became a proper
Exactly.
combined produced drug,
Mm.
right? So Very, very cool. Very nice, interesting work. I mean, I show my colors that I am a microbiologist.
Yeah.
Really
Thank you.
all right, if nobody has any more questions, I think we'll call it a Friday. We call it a Friday, and we finish today's webinar. Thank you so much for listening and being here, either live as we are happening, or maybe for a playback later on our YouTube channel.